There are more than 100 distinct modifications of RNA, 2/3 of which are methylations. This protocol provides a method for sensitive and accurate analysis of RNA methylation writer activity. This easy-to-use technique allows to quantify and to visualize methylated RNA without the use of antibodies, which are commonly prone to artifacts. Demonstrating the procedure will be Samantha Shelton, a grad student from my lab.
To begin this procedure, set up the 100 microliter RNA methyltransferase assay in a 1.5 milliliter tube on ice as outlined in the text protocol. Vortex the tube to mix thoroughly, and centrifuge at 200 times G for 5 seconds to collect the solution at the bottom of the tube. Incubate the tube at 37 degrees Celsius for 2 hours. Then, clean the reaction using column purification as outlined in the text protocol. To perform the liquid scintillation count, set up the scintillation count rack with 1 vial per sample, 1 vial for a background measurement, and 1 vial for the swipe test. Fill the vials with 5 milliliters of scintillation counting solution. Add 10 microliters of each eluted radioactive RNA sample into 1 vial. Tighten the lid and mix gently. To prepare the swipe test vials, thoroughly rub cotton swabs on all surfaces and equipment used during the procedure.